Now, inheritance style questions tend to be application based. Now, we know the basic mechanisms of both monohybrid and dihybrid crosses, and in this particular question we're going to apply that knowledge to the information that we're given. So we can see here that actually there is a diagram that we're provided with, so we are going to use this information in the answers that we give. Now we can see in the sentence at the top of the slide that there is a molecule called phenylalanine, and the question tells us this is normally metabolized into two types of molecule. One is called melanin, which is a pigment that we find in the skin, the hair and the eyes, and via a very similar pathway phenylalanine can actually be converted into a molecule called dopamine. And we know dopamine is actually a neurotransmitter in what we call dopaminergic neurons, or neurons that use dopamine as their neurotransmitter. So we know phenylalanine is actually a substrate, so we can put here substrate, and that is for an enzyme that is called enzyme A in this particular question. So we know phenylalanine is a substrate and it has a complementary shape to the shape of the active site for enzyme A. We know if there's successful collisions between phenylalanine and the substrate, the active site of enzyme A, we get an enzyme substrate complex, and the product that gets released from that enzyme catalyzed reaction is tyrosine. So we could say tyrosine is the product from the reaction of enzyme A. But tyrosine also now is a substrate for the next enzyme. So this step down here that we might want to call step number 2 will be an enzyme catalyzed reaction whereby tyrosine is a substrate, and the end product from the second reaction will be a molecule called DOPA. And again, that is a substrate for two more enzyme reactions. So we could put number 3 here, and the DOPA product, which now acts as a substrate for enzyme number 3, will form melanin, and when DOPA is a substrate for enzyme number 4, the product will be dopamine. Now underneath the diagram it does tell us that PKU, or phenylketonuria, is a condition which results from the absence of enzyme A. So we know we must always use the information provided within the question, and again we can annotate the diagram before we even start to think about writing down some answers. And we know if enzyme A is absent, this first reaction at the top here, number 1, will not take place. That's why we've got a big cross here. So phenylalanine cannot be converted to tyrosine in the absence of enzyme A. Now if there's no tyrosine because enzyme A is absent, there's no substrate for enzyme number 2, so tyrosine is not present, therefore DOPA is not made. If there's no DOPA as a product, it can't act as a substrate for the production of melanin or the production of dopamine. It then tells us in the question that people with this condition, called PKU, they are homozygous for a recessive allele, and that fails to produce enzyme A. So a person with 2 recessive alleles for the gene for enzyme A, they will not make a functional enzyme. So these recessive alleles might well be transcribed into mRNA. The mRNA might even be translated into a protein, but that protein is a non-functional enzyme, which means it doesn't catalyze phenylalanine into tyrosine. So we know healthy people with enzyme A, they must have at least 1 dominant allele for this gene. And obviously individuals with 2 dominant alleles for this gene will be able to make twice as much enzyme A inside their cells. The first question then says, use the info shown in the diagram to give one symptom you might expect to be visible in a person who inherits PKU. So all we need is one symptom. Well, we know if enzyme A is not being made, phenylalanine is not being converted to tyrosine, and therefore melanin and or dopamine are not made. So we can work out one symptom, and it's going to be to do with the lack of melanin or the lack of dopamine. So the two answers that we could put down here are an individual with PKU will have a lack of skin, eye or hair pigmentation. So they might have fair skin or fair hair. And also, because of the lack of dopamine, they might have poor coordination because of the lack of synaptic transmission between neurons, in particular between dopamine neurotransmitter neurons. So a lack of skin, eye or hair pigmentation because of a lack of melanin, or a lack of coordination due to a lack of synaptic transmission between neurons, particularly in the brain, because of a lack of dopamine. So we know that healthy people will have at least one dominant allele for enzyme A, for that gene. Now, homozygous recessive individuals with two recessive alleles will not be able to make the functional enzyme, and therefore they express the symptoms of PKU. Now, quite often recessive alleles might actually be mutant or mutations in that particular gene. And in this case, the mutation, the recessive allele, means that no functional enzyme is being made. So the question says, B, explain how gene mutations may result in the allele which fails to produce the functional enzyme. So here, this question is actually going to be about the nucleotide sequence of an allele. And if that allele is a mutant allele, it's not going to make the functional enzyme, and we need to be able to put down why. So we know a mutation is a change in the base sequence due to either a deletion, that's the removal of a nucleotide, the substitution of a nucleotide where the wrong nucleotide is inserted during maybe DNA replication, or actually an insertion of an extra nucleotide. Now we know if we have one of these mutations, it might change one or more codons. And we know an allele is a sequence of codons, with each codon specifying a specific amino acid in the end polypeptide chain. So a change in base sequence due to deletion, substitution or insertion of a nucleotide, and therefore the nitrogenous base, into the coding strand of DNA. Now this allele, this mutated allele, is the one that will be inherited in the PKU individuals. They'll have two copies of this recessive allele. Now we know if there is a mutation, one or more different codons might be formed, so the incorrect amino acids might be inserted into the final polypeptide chain, and that's during obviously translation of the mRNA at the ribosome. So we could put at the ribosome. Now the reason this might produce a non-functional enzyme is quite simply, if you change the primary structure of a protein, you change how that protein folds into its final 3D shape. So if the tertiary shape of the enzyme has been altered because of the different amino acids in the polypeptide chain, it means the active site shape is also likely to be altered. And if the active site shape has been changed, then no successful collisions can occur between the substrate phenylalanine, because it is no longer complementary in shape to the shape of the active site of the enzyme. So there's no enzyme substrate complexes formed, and therefore we could put at the end no product, i.e. no tyrosine. So this question is a very common question about why a mutant allele, or in this case the recessive allele, would not make the protein, and it's because changing the base sequence changes the codon sequence, changes the amino acids in the polypeptide chain, which changes the tertiary shape of the enzyme, so it can't react successfully with the substrate. Now question C talks about the actual monohybrid cross, whereby offspring are produced, of which some of them might have this condition called PKU. Now the actual question does tell us that a child with PKU was born to two unaffected or healthy parents, and then it asks us to complete the genetic diagram to show how this is possible. Well, we know that a child with PKU must have 2 recessive alleles for enzyme A. Now that means that one of the alleles must come from one parent and the second recessive allele must come from the other parent. Now we know that the phenotypes of both parents are healthy non-PKU for parent 1 and parent 2, because it does tell us in the question that both parents are not affected by PKU. But we know that both parents must have at least one recessive allele. And that tells us that both parents, their genotypes must be heterozygous for the mutant or the recessive allele. So we can see here, parent 1 has a genotype for this gene of a big N and a lowercase n. So that means they have one dominant allele and one recessive allele. And the second parent, they're exactly the same. They're heterozygous, one capital big N, which is the healthy allele, and one recessive lowercase n, which would be in this case the mutant allele. Now, because they are heterozygous, both parents have a dominant phenotype, and that means that they can make enzyme A because they've got one healthy allele, and therefore they can actually make melanin and dopamine. So then it says the genotypes of the gametes. So we know the parent genotype is diploid because humans are diploid, and we know the gametes of both parents, whether it be the secondary oocyte that becomes the ovum of the female or the sperm cells, spermatozoa of the male, gametes are haploid. So in the gametes, because they're haploid and not diploid, they can't have 2 alleles for that gene. They must just have one allele for that gene. Because whatever chromosome these alleles fall on, for example, maybe chromosome 7, there's 2 chromosome 7s in each nucleus of the parent because they're diploid. But in the nucleus of the gametes, the ovum or the spermatozoa, there's only 1 chromosome 7. So there's only one allele in the gametes for this gene. So when we write out the gametes, we always do them in circles. So this parent here, the diploid cell, when that undergoes meiosis, we get 2 gametes and they're both haploid. So one gamete will have uppercase N and the other gametes will have lowercase n. This is the only possibility we can have for the gametes. And it's exactly the same for the second parent. So one gamete will have uppercase N, the dominant allele, and the second type of gamete will have the recessive or mutant allele. Now you always put gametes in circles because it tells the examiner that you can see very clearly which allele is inside each gamete. Now we're going to do a Punnett square. So we know for a Punnett square you have a table. Both axes of the table you have the word gametes there, so that's very important. And then you show the gametes, and you can do them in circles if you wish, but you don't have to necessarily. And when we have fertilization, obviously this gamete here, when that fertilizes with the gamete on the left hand side, we reinstate or restore the diploid number of chromosomes. So we're going to get 2 alleles here that come together on the 2 sets of chromosomes. So the genotype will be big N, and we know that genotype is homozygous dominant. So the phenotype of that offspring we know will be the dominant phenotype, i.e. with a nice healthy enzyme A. So we can label these boxes. We'll label that offspring number 1. Offspring number 2, well, we're going to have a lowercase n, which is the recessive allele, and that will combine with the dominant allele from the other gametes. So in this case, the offspring will have a heterozygous genotype, so big N and then lowercase n, so one dominant and one recessive allele. Again, they will have the dominant phenotype because they've got one dominant allele. Offspring number 3, again, there's an uppercase N, which is the dominant allele, combines with the lowercase n, which is the recessive allele. So this individual will also be heterozygous for enzyme A, and therefore they'll have a dominant phenotype. Now, the 4th individual, because the sperm cell had one allele, which was lowercase, that's the recessive allele, and the ovum had one allele, which also in this case was the recessive allele. Offspring number 4 will be homozygous recessive. Now, because there's no dominant allele here, this individual will show the recessive phenotype, and therefore this individual will show the symptoms of PKU. Now it says, what are the genotypes of the children then? Well, we can just list the genotypes underneath the Punnett square. So there's 3 different types of genotypes here. We can see homozygous dominant. We can see offspring number 2 and number 3 are heterozygous. And we can see offspring number 4 is homozygous recessive. So then the phenotypes and the phenotype ratios, well, you always put the phenotype with the ratio number. So of the 4 offspring in the Punnett square, there's 4 children, 3 of them will have the healthy phenotype. So we go 1, 2, 3. So there's 3 healthy non-PKU children, and one of the individuals, number 4, will express the PKU condition. So you can see here that's 1 of 4. So we put the ratio as 3 healthy individuals to 1 PKU individual. And then the last question says, what is the probability that a second child born to these parents will have PKU? Well, if there's another child being born to these 2 parents, there's a 1 in 4 chance that the child will have PKU, and a 1 in 4 is exactly the same as saying a 25% probability that a child will be born with PKU to these parents. So the first question that we looked at was all to do with monohybrid inheritance, i.e. the inheritance of a single gene that codes for 1 characteristic. Now this second question is actually about a dihybrid inheritance pattern. Now we know the word di suggests 2, so dihybrid, we put this here, dihybrid suggests 2 genes are being inherited. Now this will follow a Mendelian inheritance pattern if the 2 genes are on different chromosomes. So we're provided with some information in the text of the question, and it says some forms of clover are cyanogenic, and that means that these clover plants, they can actually produce poisonous hydrogen cyanide. So we're going to use this in the answer when their tissues get damaged. Now the production of this kind of toxic molecule, hydrogen cyanide, takes place in 2 steps, and each of the steps is catalyzed by an enzyme produced by a different gene. So we know straight away that there's actually 2 genes involved, and each gene is responsible for making a certain enzyme. So there's gene 1 that will make enzyme A, and there's gene 2 that will make enzyme B. Now the question does tell us that the dominant allele, so there's going to be 2 alleles, a dominant allele and a recessive allele, and it's the dominant allele that codes for a functional enzyme. So for gene number 1, the dominant allele will actually make a functional enzyme called enzyme A. And this suggests that the recessive allele for this gene will not actually make an enzyme. So we could say here the recessive allele, and we might call that lowercase a, makes a non-functional enzyme A. And then we can see, if we go a bit further down, that the second gene, it tells us that the dominant allele B will make a functional enzyme B. And then we can say, well, the recessive allele for this second gene, and we might give that lowercase b, again, that makes a non-functional enzyme B. So as long as there's one dominant allele, then that enzyme can be made inside these cells, and that goes for both enzyme A and enzyme B. So then it tells us that as long as there is a functional enzyme A, that can convert a substrate into a product. So just before we look at the answers, we can see the substrate is catalyzed by enzyme A to form a molecule called cyanogenic glucoside. Now this product from enzyme A also acts as a substrate, a little bit like we saw in the last question, for enzyme B. And cyanogenic glucoside, being the substrate for enzyme B, is complementary in shape to the active site of enzyme B, and that is then converted by enzyme B into hydrogen cyanide. And we know hydrogen cyanide is toxic. Okay, and it's produced when the tissues are damaged on the clover plants. So the first question says, what are the phenotypes? Are they cyanogenic or non-cyanogenic of the following genotypes? So the phenotype is the physical characteristic of the cell, the tissue, or the organism, and the genotype is the alleles that are present inside the nucleus of that individual. So AA, so capital A, and then BB with a capital B and a lowercase b, and it says what's the phenotype? Well, we know for this gene, the first gene, gene A, there's 2 dominant alleles. So we know that this enzyme up here, enzyme A, is going to be expressed. So we're going to get the substrate on the left here being converted to cyanogenic glucoside. So this enzyme A is functional. It then tells us that there's 1 dominant B allele, the capital B. So these cells also make enzyme B, a functional copy of that enzyme B. So cyanogenic glucoside can now be converted to hydrogen cyanide. So actually the phenotype is going to be cyanogenic, and that's the first mark point. And then it says explain your answer. So we can pop in the answer. The functional enzymes A and B are both present inside the same cells, as there are dominant alleles for each gene. There's actually 2 dominant A alleles, AA, and there's 1 dominant B allele. So cyanogenic glucoside is made by enzyme A, and that acts now as a substrate for enzyme B, which will make hydrogen cyanide. Now we can look at the next genotype. So here we've got homozygous recessive for the first gene, and that's lowercase a, lowercase a. And we've also got homozygous dominant for the second gene. So that's uppercase B, uppercase B. Now here we can work out the phenotype, because if there's 2 recessive genes, 2 recessive alleles for enzyme A, it means no functional enzyme A is going to be formed, as we can see up here. If there's no functional enzyme A, the substrate cannot be converted to cyanogenic glucoside. If there's no cyanogenic glucoside, it doesn't actually matter if there is a functional enzyme B, because there's no substrate for that enzyme to convert into hydrogen cyanide. So these cells are not going to be able to make hydrogen cyanide, and therefore the phenotype is non-cyanogenic. So that was the first mark point, non-cyanogenic. Now the second mark point is for the explanation. So it says a functional enzyme A is absent or not made, so no cyanogenic glucoside can be formed inside these cells of this organism, meaning there's no substrate for enzyme B to work on, because there is actually an active functional enzyme B, and no hydrogen cyanide is made, hence non-cyanogenic. So question B tells us that 2 clover plants, each with genotype capital A, lowercase a, and capital B, lowercase b, were crossed. Now we know if there's 1 dominant allele for gene A and 1 recessive allele, that's heterozygous for gene A. And if there's 1 dominant allele and 1 recessive allele for gene B, this individual is heterozygous also for the second gene, gene B. So we can use the general setup for a genetic cross, and we can show the genotypes of the offspring that would be produced. So we know that we always start with the parental phenotype. So we can jot that down. There's cyanogenic parent 1. So that's a normal clover plant, cyanogenic. And there's a cyanogenic phenotype, heterozygous genotype for parent 2 as well. And we know, as it says in the questions, that the genotypes are both heterozygous for enzyme A and enzyme B. So these individual plants both make inside their cells a functional enzyme A and a functional enzyme B. Now when we write the gametes for each parent, we're going to put the gametes in circles. So we know that the parent genotypes, the parents are diploid, these plants. Now the first gene, gene A, if it's diploid, will have 2 alleles, uppercase A and lowercase a, but only one of the 2 alleles can end up in the gametes because the gametes are haploid. So for the first gene, in this gamete on the left, we've got the dominant A allele, and we know only one of the 2 alleles from gene B, the second gene, can end up in the gametes because it goes from diploid to haploid. So this time we're going to put in the dominant B allele. Now the second gamete could have the dominant A allele and the recessive B allele. The third gamete could be opposite. So we could have the recessive A allele and the dominant B allele. And the fourth gamete could have both A recessive allele and B recessive allele inside it. Now because the genotype of parent number 2, so here we put parent 1 and here we put parent 2, is the same, the gametes being produced by parent number 2 will be the same as we've just seen. So uppercase A, uppercase B, dominant A, recessive B, recessive A, dominant B, and recessive A and recessive B. Now when we draw the genetic cross, we need to do the Punnett square, and this time we're going to have 16 squares, so we can actually number all 16, and that's actually because for each parent we've got 4 different possible types of gametes. So if you times 4 by 4, you end up with 16 possible offspring genotypes. So always label your Punnett square with the word gametes. That's absolutely essential. Then you put in your gametes. So if you want, you can actually even circle them. So from parent 1, we've got the 4 gametes. From parent 2, we've also got the 4 gametes. When we get successful pollination and therefore fertilization, we restore the diploid number of chromosomes. So for each gene, we're going to have 2 alleles in the offspring, and then we can just put in the correct combinations. So if we look at this gamete here, and then we look at the other gametes on the top, we're going to have 2 dominant A alleles and 2 dominant B alleles in the offspring, and then we can just complete the Punnett square for all of the crosses. Now once you've done this and you've double checked it, we can then look at the phenotype ratios of the offspring. So any genotype with one capital A or dominant A allele and at least one dominant B allele will have the dominant phenotype. So we can jot this down, and that will mean they will be cyanogenic. So let's just cross off which ones will be cyanogenic. Well, we can see offspring 1 will be, offspring 2 will be, offspring 3 and offspring 4 will be. Offspring 5 has at least one dominant A allele and one dominant B allele, so that will be cyanogenic. Offspring 6, it does have homozygous dominant genotype for the A allele. However, it does not have a dominant B allele. It has 2 recessive B alleles, so that will not be cyanogenic. It'll be non-cyanogenic. Offspring number 7, it has one dominant A and one dominant B allele, so that will be cyanogenic. Offspring 8 will not. Offspring 9 will be, as will 10. Offspring 11, it is homozygous recessive for the A gene, the first gene, so that will not be cyanogenic. Offspring 12, again, will not be cyanogenic. 13 will be, 14 will not, 15 will not, and obviously 16 will not, because that does not have a functional A enzyme or a functional B enzyme. So we can put in the phenotype ratios then just by counting up what we've got. So if we count the ticks, it's 1, 2, 3, 4, 5, 6, 7, 8, 9 ticks. So there's 9 cynogenic offspring for every 7 non-cynogenic offspring. And then the question at the bottom says, what is the expected ratio of cynogenic to non-cynogenic plants? So we can quite simply just put that down. It's 9 cynogenic to 7 non-cynogenic. Now we normally actually give a ratio to 1. So this is the same as saying 9 divided by 7 to 1, which comes out as 1.29 cynogenic to 1 non-cynogenic. So it's always good practice to put your ratio 2 to 1. So question C is about allele frequencies, and it's going to ask us about how allele frequencies can differ under different environmental conditions. So it gives us some information at the start of the question, and it tells us that cynogenic clover plants are toxic to slugs. Now we know from the previous question that cynogenic clover plants will have the genotype A, which is homozygous dominant for gene A or gene 1, and B, so that will be homozygous dominant for gene B or gene 2. We also know that the heterozygotes for both gene A, so that will be dominant and recessive alleles, and also the heterozygotes for gene B will also be a genotype that gives rise to a phenotype that is cynogenic. Now it then tells us that slugs are actually quite common at low altitude. So we're going to start to jot this information down to make sense of the question. So we can say here cynogenic clover plants. They are toxic to slugs. Slugs we find at low altitude. But then it also tells us in the same question that not at high altitude, so we don't find slugs at high altitude where the climate is colder. So we could put in brackets here, no slugs higher up where the climate is colder. Then it says when the cells of cynogenic clover plants freeze, the hydrogen cyanide is released, which kills the cells, and as a result the plant dies. So we can add another bit of information here, and we can say, so cynogenic clover plants they freeze at high altitude where it's colder, and then the plant dies. Then it says the frequencies of the alleles for both cynogenesis and therefore non-cynogenesis also, which will be the recessive alleles, are different in clover plants grown at high and low altitudes. So the first question says, how would you expect the allele frequencies to differ? And this refers to at low altitude and high altitude. So we're going to look at the selection pressures. So this part of the question is to do with variation evolution. Now it tells us that cynogenic clover plants, they're toxic to slugs, and slugs are more common at low altitude. And we know at low altitude the temperatures are warmer. There's no slugs at the higher altitude, and we know at higher altitudes the environmental conditions are actually much colder. But at high altitudes, the cynogenic clover plants freeze, and that causes the hydrogen cyanide to be released, and it kills the actual plant itself. So we could make an assumption that there will be a higher frequency of dominant alleles at low altitude. So for example, the genotypes that we've written down here. So these genotypes are going to be much more frequent, okay, or higher frequency at low altitude, the dominant allele A and capital B. And there'll be a low frequency of dominant alleles at high altitude. And we know at high altitude there are colder temperatures. We know the cynogenic plants will freeze. It releases the hydrogen cyanide that kills the plants. So having dominant alleles, so capital A and capital B, at higher altitudes is not actually a selective advantage, because this genotype, the dominant genotype at high altitudes, will actually kill the cells and the plants will die. So having this dominant genotype for both gene A and B is actually a success, or allows the plants to successfully survive at low altitudes and therefore reproduce, because actually the clover plants are toxic to slugs, so they tend to not get eaten by the slugs. So that's going to be a selective advantage at the lower altitude where the temperatures are somewhat warmer. Right, the next question says, use the info given to explain how the different allele frequencies might be the result of natural selection. So again, we're going to use the term selected against if the environmental conditions select against a certain allele type, and we're going to use the term selected for if the environmental conditions select for a certain allele type. So there is selection against cyanogenic phenotypes, and therefore the dominant alleles A and B at high altitude, because hydrogen cyanide is released. There's selection against the non-cyanogenic phenotype, and therefore the recessive alleles, lowercase a and lowercase b. And this is going to be at low altitude because slugs eat those plants that are not making hydrogen cyanide. So if you get the recessive alleles, homozygous recessive for both gene A and gene B, in these clover plants at low altitude, the plants are not toxic to slugs, the plants will get eaten by slugs, the plants will not be able to survive, and they're going to be therefore the alleles selected against. So these are the recessive, so we put there recessive alleles. Now the first mark point up here, for which we gave a mark, was we'll get selection against cyanogenic plants, their phenotype at high altitude. So actually the dominant alleles are selected against at high altitude. There you go. So this is mark point 2 here. Now the third mark point then, we know slugs are going to eat more non-cyanogenic plants, which is going to alter their allele frequency at low altitude. So these are going to be selected against. Now when you get to variation evolution, it's always very important to jot down what is the selection pressure. So at low altitude, the selection pressure is the slugs eating the non-toxic or non-cyanogenic plants. So that's mark point 3. Right, at low altitude, more cyanogenic plants survive, and therefore they can reproduce. So they pass on the beneficial alleles, which in this case are the dominant alleles. So we know natural selection is all about alleles being selected for and passed on to their offspring if these individuals with that phenotype can survive and therefore reproduce. Mark point number 4. There's greater survival of these plants, and it means, like we just said, if they can successfully reproduce, they're much more likely to pass on these beneficial dominant alleles at low altitude, and therefore the alleles end up in the next generation of these plants. So that's mark point number 5. We could also say a higher frequency of dominant alleles in the next generation is going to occur at low altitude, and a lower frequency of dominant alleles will occur at high altitude, and that is because at the high altitude the recessive alleles are the ones being selected for by the environment, and that is because at high altitude the temperatures are colder, the cells of these cyanogenic plants tend to freeze, and therefore the plant dies.